Assalamu alaikum, ladies and gentlemen, and a very good afternoon. I am Zunaira Said. I warmly welcome you all on 7th International Conference on Applied Zoology 2024 on the behalf of Organizing Committee ICAZ. I will be the moderator of this session, which includes talks from the fields of drug discovery, environmental biology, and diabetes. Let's we start our session from the talk of Dr. Muhammad Kabir from Thal University, Bhakkar on the topic of photoremediation, a sustainable environmental technique for air pollution resulting in climate change. So please come and educate us on the topic.
Bismillahirrahmanirrahim. Respected chair, co-chair and audience, Assalamu alaikum. I am Dr. Muhammad Kabir, serving as an assistant professor and in charge at Department of Biological Sciences, Thal University, Pakistan. First of all, I say my thanks to the organizing committee, the Applied Zoological Society of Pakistan and University of Sargodha, who gave me a chance of oral presentation in this an international conference, 7th International Conference on Applied Zoology 2024. Audience, as we know that environment is very important component for existence of life on the surface of Earth. So the topic of my presentation is the phytoremediation, a sustainable environmental technique for air pollution resulting in climate change. And the presentation layout includes the problem. As we know that research starts with a problem and ends with the solution. So the problem here which I want to address is the air pollution resulting in climate change, and its solution is the plantation activities. Then what is phytoremediations, climatic changes, importance of tree for sustainable habitat, pollution, pollutant type and issue, causes and impacts of air pollution, importance of a tree, solution for pollution, plantation activity in Thal University Pakistan environmental calendar, and some quotation related to the environment, conclusion and recommendation, selected certificates, and at the end I will show you some of the publication 2024. Here you can see in the slide what is the phytoremediation and how it can be used for the treatment of the water. As we know that soil, air and water are most important component of the environment. And without water, plants cannot show their proper growth and development. And without the air and water, living organisms cannot exist on the surface of Earth. So water and air are the most important components of the environment. And as we know that soil also acts as a basic medium for the plant growth. Here you can see that in the water reservoir, when pollutants are being added by the different anthropogenic activity, this polluted water is being changed characteristics of the water by bonding with some important elements of the water, and then this water passes into the soil, and from the soil it is taken up by the plants through its roots, and then within the body of the plant different phytoremediation technique can be used by the plants for the cleanness of the water, and this water can be checked by the laboratory, and then clean water can be used for the different purposes. It means that plants are very important regarding to the clean environment. Plants act as the natural lungs of the universe because they take up carbon dioxide from the environment, and as a result they give out the oxygen which is necessary for existence of life on the surface of Earth. So plants are important regarding to all the three components of the environment, that is air, water and soil, and they act as a phytoremediator. What is the climate change? As we know that this word is very common in our society, that climate is being changed. We must firstly know that what is the climate. The climate is an important environmental factor of an area and country and even the whole world. The changes in environmental factor of an area over a long period of time constitute the climatic changes, and these climatic changes are affecting all the hydrological cycle, wind pattern, distributions of rainfall, growth and development of the plants, and even whole food chain on the surface of Earth. As we know that plants are the producer on the surface of Earth, but plants are also threatened by the different pollutants which are being discharged into all components of the environment, and mainly due to the anthropogenic activities. It means that human is primarily responsible for production of different pollutants into all components of the environment. According to the UNCC, which shows that Pakistan is one in the list of the top 3 countries mostly threatened by the climatic changes. So what is the pollution, its type, pollutant, and what is the issue behind this? As we know that pollution is an undesirable change in physical, chemical, and biological characteristics of the air, water, and soil that may harmfully affect to the living organisms. And the substances which cause the pollution into all components of environment are known as the pollutant, or we can simply say the substances which cause the pollution are known as pollutant, and the residue of thing we make you and throw away into the environment are also known as the pollutant. So what is the issue behind this? The issue is that we use the resources, that is air, water, and soil, without their consequences. It means that human is most species on the surface of far regarding to degradations of the environment and resulting in the different types of pollutant into all components of the environment, mainly air, water, and soil. So the main causes of the pollution includes the overpopulation, urbanization, industrialization, transportation, deforestation, and so on. And its effects include the biodiversity loss, floods, landslides, soil erosion, endangerment of the species. And as we know that the solution are at very low level as compared to that of the causes and the effect. And so the solution includes the control of the population explosion, stop deforestation, promote air reforestation, fine for illegal dumping, strict government regulation, and so on. Here you can see in the picture that causes and impacts of the air pollution. It is our common practice that we carry out the threshing of most of crops into the open area, and this activity releases a large number of the aerosol and some other particulate matter into air that ultimately pollute the air, reducing the air quality. Our different transportation activities are also produce different types of pollutant into the air, and our bricklaying also producing the different gases into the environment. And it is our common practice that after cultivation of the crop we burn the open stalks into the field, which also produces different types of the pollutant and toxic gases into the air. So all these human activities are responsible for production of different types of pollutant into the air. Our industrial activity is also releasing different types of pollutant into the air, which ultimately pollute the air and reduce its quality. As we know that air is the most important component of the environment for existence of life on the surface of Earth. It is our common practice that most of the industrial waste dumped outside the industrial wall, and after their burning, different types of the pollutants are being released into all components of the environment, including the air, water, and the soil. So human activity is not promoting the friendly environment, but we are degrading the environment. It is our common practice that we burn and cut most of the tree for our different purposes. We should know the importance of the tree, how trees are important regarding to the environment, and then we should carry on the deforestation activity. As we know that Pakistan required 33% forest area, but according to the latest report of the UNO, this area has been reduced only up to the 2%. So what is the importance of the tree? A tree that lives for 50 years generates a $US worth of oxygen, recycles $US worth of the soil fertility, facilitates $US worth of the soil erosion control, creates $US worth of the air pollution, and provides $US worth of the shelter for insect birds and animal. Beside it provides flower, fruit, and seed. Our net loss is worth more than 57,381 when tree fall are affected by the environmental pollution. Therefore, a humble request to all the humanity on the surface of Earth is that think before you cut a tree. You can see the plantations activity in our Thal University Pakka, that we have planted a large number of trees at our entrance gate. We also have the earthen pots and some large number of the tree in front of our academic block. Our student, faculty member, and staff are in different walks and talks in collaboration with the district administration to carry out the plantations activity at the district level as well as in the campus. We also have collaboration with the Pakistan Army and some other organization at the district level to carry out the plantations activity. Here you can see that environment has very important days. The environmental calendar shows the importance of the different dates regarding to the environment. And environmental calendar doesn't have only the dates, it has a weeks and the years. If we are environmentally friend, we must celebrate all these environmental events so that we can keep our environment clean and green, and we can give the sustainable environment to our next coming generation. So the conclusion is that if haphazard population growth and construction of the new industrial structure, transportation, urbanization, and deforestation go on without realizing the importance of the trees, then probably there would be more destructive environmental changes in near future, and these environmental changes are harmfully affecting the habitat of the living organism. Recommendation is that be a part of the solution, not pollution. Request to each and every participant of the conference is that each one must plant at least one. Join the green revolution and stop the pollution. Here are some of my certificate which I obtained as a research productivity award from Pakistan Council of Science and Technology in 2011 and 12, and these are the publication of 2024. And here is the end of my presentation. Thank you for your patience. If you have any question, that I am here. Thank you.
Thank you so much, sir, for sharing your valuable knowledge with us. Now, I would like to invite Dr. Shagufta Saeed from UVAS to talk on the topic of green synthesis of zinc oxide nanoparticles using Achiranthus aspera leaf extract and their potent action against the poultry. Ma'am, please come and educate us on the topic.
Assalamualaikum. First of all, thank you to the organizing committee for inviting me in 7th International Conference on Applied Zoology. I am Dr. Shagufta Said from Department of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences Lab. Today, topic I am going to discuss is green synthesis of zinc oxide nanoparticles using Acanthus aspera extract and their potent action against poultry pathogens. First of all, introduction. Poultry industry gained a very important role in economic development of Pakistan in past few years, and poultry meat consumption is progressively increasing worldwide. It reached 14.2 kg per capita per year. Mortality rate in poultry has been growing every year due to the outbreak of several diseases, and these diseases are the major drawback in advancement of poultry industry, which lessen the prime production of the flock. Most of the microbial poultry pathogens are Salmonella and Campylobacter. Microbial safety of poultry meat product is an important issue in context of increasing consumption and its production. Now, what are these? We are focusing basically on these two poultry microbes. Number one is Salmonella gallinarum and second is Salmonella enteritis. Salmonella gallinarum is a rod-shaped gram-negative bacteria. It contaminates eggs and making it potent infectious agent for humans. And second one is the Salmonella enteritis. It is also a rod-shaped gram-negative facultative anaerobe and it transmitted via improperly cooked meat, contaminated food or water. Nanoparticles and an alternative source of antibiotics. The unregulated use of antibiotics provide the impulse for bacteria to become drug resistant, and nanotechnology is becoming as an alternative source against antibiotic resistant poultry pathogens. Nanoparticles have developed antimicrobial approach to fight for treating severe bacterial infection. Nanoparticles are atomic or molecular aggregate characterized by size less than 10 nanometer. Now, what is green synthesis? It is basically synthesis of nanoparticle using plant source such as stem, roots, leaves, and flowers. Green method had progressively becoming a topic of interest because conventional methods are expensive, time-consuming, and its use of chemical compound like organic solvent as a reducing agent. Various metal oxide nanoparticles like iron oxide, titanium oxide, copper oxide have been synthesized using plant sources. So what are zinc oxide nanoparticles? Zinc oxide nanoparticles have been a keen area of interest due to their distinguished antimicrobial activity, and It is an inorganic compound prepared as a white powder soluble in water. Antibacterial activity of zinc against various pathogens such as Staphylococcus aureus, Salmonella, Bacillus subtilis, and E. coli have been reported. Acranthus aspera. It is a species plant in Amaranthaceae family. Its common is scarflower, devil worship, horseship, distributed throughout the tropical world, and its habitated dry and warm places. Its height is 1 to 2 meters, and it commonly found as a weed. Now, statement of problem. The extensive use of antibiotics has raised concern about increased resistance of Salmonella in poultry and causes serious health hazard and economic losses. Hypothesis. Zinc oxide nanoparticle produced by leaf extract of Acranthus aspera have antimicrobial potential against poultry pathogens. Objectives: synthesis and characterization of zinc oxide nanoparticles, and second one is evaluation of antimicrobial activity against poultry pathogens. Material method: first of all, we have to collect the sample of the plant, and fresh leaves of Acranthus aspera plant was collected from University of Punjab, Quaid-e-Azam campus, Lahore, and after collection, leaves was washed with the distilled water and oven dried at 60 degrees centigrade for 1 hour, and then it is grinded to make powder. Second is the extract preparation. 20 grams of powder was dissolved in a distilled water, and then it is heated for 30 minutes in a mantle, and then this extract is filtered through a filter paper, and obtained extract was stored. Preparation of zinc oxide nanoparticles. First of all, leave it Acranthus aspera leaf extracts, and then you mix it in 0.1 molar zinc acetate and sodium hydroxide solution, and then place it on a magnetic stirrer for 2 minutes. After that, centrifuge and washed with distilled water and ethanol, and placed in a hot air oven at 60 degrees centigrade for 2 hours for accurate formation of zinc oxide nanoparticles. Characterization of the obtained zinc oxide nanoparticles was done by following techniques. Number one, visible spectrophotometer, X-ray diffraction analysis, scanning electron microscopy, Fourier transform infrared microscopy, and atomic force microscopy, AFM. Evaluation of antimicrobial activity of zinc oxide nanoparticle was done against 2 strains of poultry pathogens, Salmonella gallinarum and Salmonella enteritidis. And all this work was done in the Institute of Microbiology, University of Veterinary and Animal Sciences. Method for evaluation of antimicrobial activity was done. First one was diffusion, and second one was minimum inhibitory concentration. Well diffusion method was done by the following methodology. And minimum inhibitory concentration was done by microdilution method, in which decreasing concentration of nanoparticles like 180, 60, 40, 20 microgram will be used. And DMSO was used as negative control in this experiment. And all the plates was incubated at 37 degrees centigrade for 24 hours, and MIC was incubated. Statistical analysis of whole experiment was done, and via descriptive analysis by means and standard deviation known as PSS. This was the result of UV visible spectrophotometer of zinc oxide nanoparticles that is show peak at 370 nanometer. These are the result of X-ray diffraction analysis. Different peaks show different peaks show the angles of the different functional group present in this molecule. This is the AFM structure, a atomic force microscopy structure that tells us about the size of nanoparticles. This is an FTIR analysis of nanoparticles, and it showed different peak 1, 2, 3, 4, and different bands, contractions, and bands of functional group. This is the SEM analysis, scanning electron micrograph of synthesizing oxide nanoparticles. And then antimicrobial activity against the Salmonella enteritidis and Salmonella gallinaриum, as it shown. First one is extract of Acranthus aspera when 100 milligram per ml was used. Then it showed no zone and negative control, no zone, and zinc oxide nanoparticles. When we use zinc oxide nanoparticles, it showed 28.8 millimeter zone against Salmonella enteritidis and 27.6 millimeter zone, inhibiting zone against Salmonella gallinaриum. And minimum inhibitory concentration was greater than 0.195 of Salmonella enteritidis and 0.39 against Salmonella gallinaриum. As you can see in the picture, first one shows zinc oxide nanoparticles, second show plant extract, and it does not show any zone of inhibition, and third is antibiotic status control as used as a positive control, and fourth is TMSO, in which we dissolve our nanoparticles. And in figure 2, it show against the Salmonella gallinaриum. First is circle show zinc oxide nanoparticles, second one plant extract, and third one antibiotic tetracycline, and fourth one DMSO. Its minimum inhibitory concentration of zinc oxide nanoparticles for Salmonella gallinaриum and enteritidis that was greater than or equal to 0.39 milligram and greater than or equal to 0.195 milligram respectively. Conclusion: zinc oxide nanoparticles synthesized from Acranthus aspera extract has been proved as a better antibacterial agent against poultry pathogens. These are the references. Thank you very much for attention.
Thank you so much, ma'am, for sharing your valuable knowledge with us. Now, I would like to invite Savera Dilbar from GCUF to give a presentation on the topic of single high dose streptozotocin treatment severely damages kidneys in adult Wistar rats. Please come and share your knowledge with us.
Assalamu alaikum. My name is Vera. I am from Government College University, Hyderabad. My presentation topic is single high dose streptozotocin treatment severely damage kidney in adult Wistar rats. Streptozotocin and alloxan are essential and commonly recommended laborogenic agents. Streptozotocin has the versatility to induce both type 1 and type 2 diabetes. When alloxan is primarily used for induction of type 1 diabetes, streptozotocin acts as a free radical generator, leading to DNA damage and subsequent cell death, and can be administered at either a single high dose or multiple low doses. Notably, STG is a highly selective cytotoxic agent that specifically targets and damages pancreatic beta cells, which are responsible for insulin production. Kidney. The kidney filter excess glucose from plasma utilizing renal glucose transporters located in proximal tubules of nephron to reabsorb the glucose. Objective. This study was conducted to investigate the effect of single high dose of STC on kidney function. Renal function was assessed through microscopic examination and biochemical analysis following STC treatment. Serum level of creatinine, blood urea, nitrogen, insulin, and hemoglobin were measured to understand the progression of diabetic nephropathy. Additionally, histological changes in the pancreas and kidney tissues were examined. Material and method. A total of 6 gram albino blister rats were obtained from the Department of Physiology at the Animal Research Station. Water and job maintenance diet were provided to every rat on an as-needed basis. The Government College University Faisalabad Animal Research Ethical Committee guidelines were followed in carrying out each and every experimental protocol. A single intraperitoneal injection of STC 200 mg was given to adult rats. Animals were sacrificed on multiple time points after STC treatment, 12, 24, and 36 hours. Blood glucose levels were measured by taking the tailwind using glucometer. Tissue from kidney and pancreas was assembled at the designated hours of sacrifice and it was permanently fixed in 10% formalin. Blood was taken in tubes and the plasma was obtained by centrifuging the tubes at 4000 rpm and serum sample was collected in an Pandoff container and frozen at -20 Celsius. Graphs. This graph represents the urea and burn level at different time intervals after 24 hours, 12 hours, and 36 hours. Urea is a waste product in the blood and burn test is used to measure the urea and nitrogen in the blood. Creatinine level. This graph represents the creatinine level at different time intervals 12, 24, and 36 hours. X-axis represents the time intervals 12 hours, 24 hours, and 36 hours post-STC administration. Y-axis represents the creatinine concentration in blood. At 12 hours, creatinine levels are lower compared to later time points. By 24 hours, creatinine level rise significantly, indicated the kidney filtration is becoming impaired. At 36 hours, creatinine levels continue to increase, reaching their highest point, suggesting progressive and worsening kidney damage. The increased trend of creatinine level from 12 hours to 36 hours suggests a time-dependent deterioration of kidney function due to STD-induced toxicity. Glucose level. This graph shows blood glucose level over time measured at 3 intervals 12 hours, 24 hours, and 36 hours. Y-axis represents glucose concentration ranging from 0 to 700. X-axis represents the time point after treatment on intervention after STC administration 12 hours, 24 hours, and 36 hours. At 12 hours, blood glucose levels are quite low. At 24 hours, blood glucose levels spike significantly. By 36 hours, blood glucose levels remain elevated, indicated the hyperglycemia persists. This graph effectively demonstrates the progression of hyperglycemia in animal model following STC administration, providing a clear timeline of how STC-induced diabetes developed over 36 hours. HP level. HP level almost remain unchanged after 24, 12, and 36 hours. This is the histological pictures of kidney. Kidney damage. Kidney tubular necrosis, damage to renal tubular cells leading to cell death. Glomerular damage, injury to glomerular capillaries, mesangium, and podocyte. Interstitial inflammation, infiltration of immune cells. Apoptosis means programmed cell death in renal cells. We can see here histological pictures of pancreas damage, to pancreatic beta cells, inflammation of pancreatic isolates, programmed cell death in pancreatic beta cells, reduce insulin staining. Conclusion: blood urea nitrogen and serum creatinine levels were not comparable between treated group and control group. Microscopic examination revealed signs of tubular or glomerular damage. The currently study revealed that single high dose STD induced dose impacted the kidney. Thank you.
Thank you, Svera, for sharing your words with us. Now, I would like to invite the fourth presenter of the session, Kainat Arshad from GCUF, to give a presentation on the topic of repeated low-dose alloxan treatment fails to induce tubular damage in adult Wistar rats. Please come and share your words with us.
My name is Kainat and I am from Physiology Department, Varun College, University of Pakistan. My presentation topic for ICAZ-2024 is repeated low dose of alloxan treatment fails to induce tubular damage in adult Wistar rats. Alloxan kidney filter out excess plasma glucose. Kidney play a crucial role in excretion by filtering out excess plasma and the waste products in the form of urine. Alloxan, a beta cell cytotoxic chemical, is commonly used to induce hyperglycemia in rodent model. Alloxan induces the formation of reactive oxygen species within beta cells, and reactive oxygen species are highly reactive molecules that can damage cellular structures, including DNA, protein, and lipids. Alloxan, it is commonly known as allox, and its complete name is alloxanum, and it is an organic compound that is synthesized in the laboratory. And the sphere of action of alloxan are pancreas, liver, kidney, blood, and lung. But the main organs that are mainly effective and affiliated by the alloxan, these are pancreas, kidney, and liver. The necrosis occurs specifically in beta cells due to their selective uptake and accumulation of alloxan compared to the cell. Alloxan is a mimic glucose. It mimics glucose and it is a glucose analog. When glucose is uptake by the glucose transport cell, the alloxan is also this is also taken up by the glucose transport cells on the pancreas, and when they enter, they are when they enter pancreas, they selectively damage beta cells. When they will damage beta cells, they will cause the production of reactive oxygen species that will ultimately cause the production of oxidative stress. And oxidative stress, these itself cause halt many physiological functions, halt many physiological functions. And when the beta cells will destroy, they will be unable to produce insulin, and there will be no insulin. Condition occurring to the body called type 1 diabetes. The present study was designed to study the impact of multiple repeated low dose of loxone on the kidney. Our aim for this study was to, because I have mentioned before that the loxone have mainly effect along kidney, liver, and pancreas. Our aim was to investigate that how much the repeated low dose of loxone have damage to the kidney, pancreas. So we evaluate renal function post-loxone treatment through microscopic examination and biochemical assessments, and measure serum levels of creatinine, blood urea nitrogen, insulin, hemoglobin to understand the progression of diabetic nephropathy, and examine histological changes in the pancreatic and kidney tissue. We took material method. We took a loxone from the health biology lab of Government College University, Faisalabad, and the rats, 6 rats. We made 6 rats from the research station of the Department of Physiology, 180 to 200 gram rats that weighed 180 to 200 grams were obtained, and they were acclimated. They were acclimated for 1 week and at temperature of 26 degrees Celsius and 40 to 60% humidity, and water and maintenance was provided to every rat. And intraperitoneal injection of loxone 40 mg was used to induce diabetes for 4 days in a row, and nicotinamide was also injected of post-loxone treatment, and after 24 hours, the blood glucose level was measured. We measured blood glucose level by pricking the tail vein using glucometer, and rats having blood glucose level higher than 180 grams were considered diabetic. And the animals were sacrificed on multiple time points at post-loxone treatment in 12, 24, and 36 hours. The blood samples were assessed for CBC, serum creatinine, blood urea nitrogen, and the kidney and pancreas were also collected and fixed for histological analysis. The blood urea nitrogen and serum creatinine levels were comparable between the treated group and the control group. Pause. As I have mentioned that these the results were comparable because the blood there, as you can see that there is a spike in the blood glucose level, but the plasma level of blood urea nitrogen and urea and the creatinine levels, these all are comparable. There is no big difference. So this is the histological microscopic picture of kidney. As you can see that there is a small amount of damage to kidneys and pancreas. The pancreas and kidneys are not effective and their function is intact. The repeated low dose of loxone do not cause huge damage to the pancreas and kidneys. The blood urea nitrogen and serum creatinine levels were comparable, and the microscopic examination reveals no sign of tubular damage. Uh, the current study reveals that repeated low dose of naloxone does not impact kidneys. Thank you.
Thank you so much for sharing your valuable knowledge with us. Now, I would like to proceed towards the fifth and the last presenter of this session, Kainat Arshad from GCUF, to give a presentation on the topic of minimal renal impact of repeated low dose streptozotocin in adult visceral rats. Please come and share your knowledge with us on this topic.
Assalamu alaikum. My name is Zinat Nasi. I'm here to represent on the International Conference of Applied Zoology 2024. The topic I am from Department of Physiology from Government College University of Faisalabad. Today I am here to represent my thesis research topic minimal renal impact of repeated low dose streptozotocin in adult blister rats. First of all, I'm here to explain about the streptozotocins. Streptozotocin and alloxans are the diabetogenetic drugs. These drugs are given to the rats to cause diabetes. Streptozotocin can be used to induce either type 1 diabetes or type 1 or type 2 diabetes, where alloxan is commonly used to induce type 1 diabetes. It is highly selective cytotoxic agents that specifically target enzymes and diabetes. Low dose injections. Kidney filter out excess plasma glucose. Renal glucose transporter present in the proximal tubules of the nephrons and employed for the uptake of glucose. So we can discuss here about how streptozotocin enter into the kidney. Low dose of streptozotocin can enter the kidney throughout the bloodstreams after being administered, usually are injections. STZ is typically given through intravenous or intraperitoneal injections. It enters the systematic circulation and is distributed throughout the body. Once in the bloodstreams, STZ travels through the circulatory systems and reaches various organs, including the kidney. The kidney filter bloods to remove waste products and regulate fluid balance. STZ being a small molecules can be filtered through the glomeruli. Glomeruli is the filtering unit of the kidney. After filtration, some of the STZ can be absorbed in the renal tubules depending on the concentration and condition within the kidney. In the kidney, streptozotocin can selectively enter renal cell, particularly those in the proximal tubules. The exact mechanisms involve specific transporters which can facilitate the uptake of the drugs into the cells. Once inside the renal cells, mainly the high dose of streptozotocin can exert into a toxic effects, contributing the oxidative stress and inflammations, which may lead to the damage and dysfunctions over the times. The main objective of this research is to present study was designed to study the impact of multiple repeated low dose of streptozotocin on kidney and to evaluate the renal functions using microscopic inspections and biochemical analysis, and also assesses the serum levels of creatinine, for the development of diabetic nephropathy to look at histological alterations in the tissues of the pancreas and the kidneys. Then there is a material and method. 6 viscera rats, adult viscera rats, and their weight are around about 180 to 200 grams, which we were obtained from the Animal Research Station Department of Physiology. Their rats were given CMP diets and waters. All the experimental protocols were performed taking into the direction of Animal Research Ethical Committee, Government College, University, Faisalabad. Diabetes was induced by administering intraperitoneal injections of low-dose streptozotocins was given 35 mg over kg for 4 days consecutively. These rats were given intraperitoneal injections of fresh diluted nectanomide 15 minutes after receiving intraperitoneal streptozotocin treatments. Then there is a 24 hours after last injection, blood glucose levels were measured by pricking the tail vein using glucometers. Rats having blood higher glucose level than 180 were considered diabetic. Glucose was mainly monitored by glucometers. All the rats were humanly euthanized and blood and tissue samples were collected for further biochemical and histological analysis. Then pancreas and kidney tissue samples were collected in 10% formalin solutions. These tissues were collected for the histological examinations. In histological examinations, further many stages are involved, in which first we collect the samples of the desired tissues, then there is a fixation, washing, dehydrations, then there is cleaning, after embedding, sectioning, mounting, and staining. These methods are for the histological examinations. Then we get a result. Here is the plasma creatinine levels of control and treated values, and hemoglobin. It is the mean plasma level of blood, urea, nitrogen, which is also called blood and urea. Mainly in this topic, we can, in this research, streptozotocins can cause diabetes, but it cannot affect on the kidney. The test results were not changes. Here is the slides of blood glucose level, in which blood glucose levels are rises above to 180, and these rats are also diabetic. Histological picture of control and treatment groups. Then there is no effect on these pictures. In conclusion, blood, urea, nitrogen, and serum creatinine levels were comparable between the treated groups and the control groups. Microscopic examination revealed no signs of tubular and glomerular damage. Study reveals that the low-dose streptozotocin did not impact on the kidney. Thank you for listening.
Thank you so much for sharing your valuable knowledge on the topics with us. This was our last session and the last presentation of this session. For anyone who missed the conference or their relative sessions can educate themselves with these links. The link of each session is provided separately for your convenience. We hope you enjoy themselves and educate yourselves with them. Thank you so much.